Well, welcome to today's presentation. We wanted to tell you what's up at the ALS Hope Foundation Dr. Sinnott Research Lab, because we've been active despite COVID-19 and keeping things moving. We're proud of the lab, and we really appreciate the gift that the friends of Bob Sinnott have given us in order to keep our lab really moving forward at a rapid pace. And we feel like we want to tell everybody what's been going on, and especially for Dr. Sinnott, have him hear how his lab is really being put to good use. And I'd like to introduce our lab staff along with me. We have Nicole Cacavo, John Fury, and of course, Dr. Bill Alexander. And they're going to help with our presentation today. And you're going to hear from all of us a little bit about what we're doing. And part of course, the gift from the Sinnott family and friends is to buy equipment so that we can really do things more efficiently, including we're getting a new PCR machine called the qPCR, and we have our Luminex, and we've gotten some freezers and some other equipment. So we thank you for keeping this lab moving forward and humming despite everything going on. And so some of the things we're going to talk about our work looking for genes that modify the severity of ALS in mice, and about our efforts to identify those genes and then translate them to people. And we've actually developed a consortium of investigators across the country who all met in September to really talk about the information we've gotten from the mouse. How do we move that to people and study people? Because these modifiers, these genes that alter disease, can serve as targets for treatment and also help us better understand ALS and motor neuron disease, and even provide biomarkers of disease and different types of disease. You know that ALS is heterogeneous, and there's actually a lot of different flavors. People all look like progressive motor neuron disease. Their motor nerves die, but the cause underlying it may be different between people. And we know now that, for instance, there are more than 30 genes that, when damaged, can cause disease. That isn't one disease, that's 30 diseases. And in people that don't have the disease in the family, that aren't clearly genetic, we suspect that there is a genetic background that determines how severe the disease will be and interacts with the environment, and that it's over time, with the genetic burden and exposure, you then develop ALS or motor neuron degeneration. So understanding how our genes modify disease is going to be really important. We also have developed an animal model, a very low expressing model of ALS. We're not going to talk much about that today, but this model is much more like people. It gets disease very late and doesn't always manifest the disease. And so it's a good model to explore the effect of different environmental insults like heavy metals or trauma on disease expression. We're going to talk a little bit, though, about oxidative stress in ALS, and John is going to tell you about that. We're looking at, do people with ALS have measurable increases in oxidative stress? Certainly, that's been shown in clinical populations that ALS may actually drive disease. It does it in the animal model, and in some folks it appears to drive it. And in fact, we now know that radicava likely is a free radical scavenger, reducing oxidative stress. And so we're looking at the ALS population, and we're looking at the effect of, and asking questions like, is there increased oxidative stress? Does it differ with different flavors of ALS, people who are predominantly bulbar, women versus men? And does radicava actually have an effect on that level of oxidative stress? And finally, we think it's really important for research to maintain tissues and autopsy material, because when we have a theory, we need to bring it not into mice, but into people and see if that theory actually pans out when we look at people with ALS. So these are just some of the things that we're doing in the lab, and we're going to tell you a little bit about each of those in a little more detail. And I'm going to turn this over to Bill right now, Dr. Alexander, who is going to tell us about our genetic modifiers in mice. Bill?
Okay. So as you guys probably know, the SOD1 gene was the first gene identified to cause ALS in humans. And not much after that, genetic mice, which were transgenic for the human mutant gene, were also shown to get motor neuron disease. About a couple of decades ago, it doesn't seem that long, but it is, we noticed that different strains of mice get motor neuron disease at different levels. The C57 black mice live longer and have a less pronounced motor neuron death, while the SJL mice have a shorter lifetime and get sicker faster. And also their offspring, the F1 generation, if you cross them, they're intermediate. We went ahead and looked, along with the people at Jackson Lab, to see where in the genome of the mouse were the genes that were modifying motor neuron disease in the SOD mice. And a major quantitative trait loci was found in mouse chromosome 17, where many of the genes that modify disease were located. They also found QTLs on chromosome 4 and 7, but the one on 17 had the much stronger, what they call an LOD score. So it had the major effect was located in this area. They went ahead and did two different strains of mice. We looked at crosses between B6 and SJL. They looked at crosses between B6 and ALR mouse, and they found that the same region on chromosome 17 had all of the modifying genes that were altering motor neuron disease in mice. Okay. In order to investigate where these genes were located within chromosome 17, interval-specific congenic mice were constructed. These are kind of a chimera. They have the genetic background of a mouse that is mainly resistant to ALS, except for the piece on chromosome 17 from mice that are more susceptible. Okay, next. We've constructed two types of interval-specific congenics. They were all on the B6 background. Some had genes from a NOT strain, and others had genes from an SJL strain. For the NOT strain, these animals lived about 17 days less than the normal B6 mouse if they carry the complete interval between 0 and 70 megabases on chromosome 17. So by breeding, we started creating interval-specific subcongenics with smaller pieces of the chromosome. The 0 to 24 mouse also moved phenotype and they lived less, whereas the 0 to 9 did not. So it leads us to believe that all of the genes that modify survival in these animals are between 9 and 24 megabases on chromosome 17. The ones with the SJL interval was from 17 to 52. The 17 to 52 didn't seem to do much for survival, but when you start splitting it, you can see that the more proximal end of it, 17 to 46, the animals live longer. And the distal segment, 45 to 52, the animals live less. So in here, we have two areas of chromosome 17 that modify survival, and they do it in opposite directions. So if you have the whole interval, it doesn't do anything. If you split it, the distal segment makes the animals live longer, shorter. The proximal segment makes the animals live longer. So that that gives us 3 segments of chromosome 17 that modify survival. 0 to 24 makes them live less. 45 to 52 makes them live less. 17 to 46 makes them live more. We also try to see if these intervals modify the survival on a different model of motor neuron disease. So these mice, same as the SOD mice, are on a B6 background, okay, with the human dynactin mutant gene inserted. And the animals also showed the same kind of pattern that the SOD mutant mice do. In the SJL background, they live less. You can see the curve on the left in red. If you put it on the B6 background, they live longer, which is blue. If you breed them together, the F1 lives in between, which is gray. And kind of a little different from the SOD mice, if you put the 17 to 52 interval in there, they live longer. So the main result of the SJL is the positive modifier in these guys. And we published that in 2015, so we've been working on that for over 5 years with the dynactin mice.
Well, that's, of course, Bill, because they live forever.
They live forever, yes. Yeah, the SOD mutation, I mean, it's kind of overloaded. The mice have 25 copies of the human mutant gene, and they live for 5 months. In the dynactin mutation, if you only have 1 copy, they live for a year and a half. If you have 2, they live for about 11 months. So it's a much slower progress working with dynactin mice than working with SOD mice. So this is kind of a summary. It's kind of a complicated if you look at the little table on the left, but essentially what it says is that there's a deleterious modifier between 9 and 24 that modifies survival only on the SOD mice. It doesn't do anything for dynactin. There's a deleterious modifier that makes both the dynactin mice and the SOD mice also live less. So that is a shared modifier that modifies at least 2 different types of ALS in mice. And then the beneficial modifier is somewhere between probably 17 and, well, 24 and 46. It is both NOD, in the NOD mice between 24 and 70, and in the SJL between 17 and 46. So if it's the same modifier, it's somewhere between 24 and 46. And this is just a very complicated slide that you should just kind of look at it very quickly. What it shows is all of the genes that are in the interval between 9 and 24, and the 3 genes that are our top candidates in that area. These are genes that have mutations in both NOD and SJL that are deleterious. Okay, our top candidate is probably SOD2. Okay, and that gene has been shown to also in other experiments with what they call not SOD2 knockout mice. They show that these animals also modify disease in the SJL and NOD and B6 SOD1 mutants. So our top candidate is SOD2, and we're conducting a series of experiments right now to see the expression of SOD2 on our interval-specific congenic as well as our B6 mice.
And so that information is being taken to human populations and looking at whether or not there's differences in people who have lived longer or shorter, if they have particular flavors of this gene. And those flavors are, called polymorphic changes. So the good thing about this particular mutation is that it's very, very penetrant. However,
it seems to only apply to SOD-ALS in humans. So it may not be a modifier that be a top candidate for any other type of ALS in humans. But we will see. We don't know that to be sure, but it's beginning to look that way.
Thanks very much, Bill. And now Nicole's going to tell us a little bit about what she does in the lab. And she's our right hand in the lab, does all the handling of the animals, mating, and so forth. Go ahead, Nicole.
Sure. So in regards to our mutant mice in particular, I'm looking at the SOD1 and the dynactin mice in particular. What I usually do on a daily basis is give them a basic assessment. I grade them on a 0 to 3 scale. So I will take certain activities such as splay, ladder up, and ladder down. When I look at their splay, it's about how far they can remove their hind legs away from their body. 0 being they can spread it out and they're pointed. It's pretty far away from their body. That usually is seen between days 40 and 60 when they're very young. And then if they are scored a 3, they can barely move their legs from their bodies. And then I also look at ladder up and ladder down. And I will score them on a 0 to 3 scale. And on that 0 scale is they can climb the ladder and go down the ladder with no problem, alternating their legs without any tripping or sliding. And then a 3 would be they can barely climb the ladder, or they can't pull themselves up, and going down the ladder they'll fall or they'll just slip all the way down. So that's pretty much what I do with the mice. Again, like what Terry said, I also handle the breeding with the mice down there. And I use our qPCR machine to find whether or not they're mutants or not. So that's pretty much what I do. And I'm still going in and finding that out on a weekly basis. So it's been really good.
Nicole also is responsible for doing what we call the Luminex, which is the way we determine how big a piece of extra DNA is inserted into the mouse, how much of that other DNA from the other strain. Remember, they're all black 6, except for one little area on chromosome 17 that belongs to SJL or not. And we have to know exactly where it is, because when the animals mate, those intervals get smaller and smaller due to a phenomenon called crossovers that everybody has. We as people have crossovers, and that gives us variability in our genome, but we actually are banking on those crossovers so that we can get smaller and smaller intervals and narrow the region that contains those possible genes. And now we're going to hear a little bit from John, who's been working in the lab and doing some of the work on oxidative stress and making sure that our tissue bank is up to snuff. Why don't you go ahead, John, and tell people a little bit about what you do in the last few minutes.
Right, thank you. Hi, I'm John Fury. I also work in the lab, and we're looking at some oxidative stress studies. So we're working with 5 other sites. So what oxidative stress is, really quick in a nutshell, is you have these angry guys down at the bottom that have unpaired electrons, and they tend to attack and disrupt cellular processes in cells that they're involved with, and that could lead to cellular damage and cell death. So then that's when the good guys hope to come in. They're the antioxidants or the free radical scavengers. They look to donate electrons or react with these reactive oxygen species, free radicals, in hopes to combat that damaging effects of those. So the goal is to keep an appropriate balance of these in our body. So this can be seen in ALS. It has been indicated, and damage can be caused by these reactive oxygen species, and this may be causing damage to the motor neurons, and this can be associated with those functional changes that we see in the disease, weakness, atrophy, and so on. So that brings us to this study that we're doing, and the drug Radicava that we all know is thought to act as a free radical scavenger, so it combats the damage caused by these reactive oxygen species seen. and this is the good guy with the big smile on the last slide, right? So it's hoping to come in and react with these reactive oxidative species to sort of slow down their damaging effect and prevent slow down their effect on these motor neurons. So like I said, we're collaborating with a few other sites as the lead and pals who are and are not taking that drug Radicava, and we want to see, does that drug in fact work as a free radical scavenger. We also want to look at do the different phenotypes show anything that is characteristic of being a stronger responder to the drug. That's what we hope to find out with this study.
And by phenotype, what we mean is female versus male, somebody who has a rapid progression or slow progression, and also whether the onset was with speech and swallowing or with extremities. So these are different phenotypes, and also whether or not somebody's what we call predominantly upper motor neuron or lower motor neuron, or quote classical ALS. And we think these may have different mechanisms or respond differently to treatment strategies. And so we want to explore that in this study. And John, go ahead, tell everybody a little bit more about the tissue repository.
So, we may be unable to collect new samples in this current environment, but we're still keeping the research very much alive. So we're taking this time to ask some pretty good research questions on the samples that we have collected previously. So in our tissue bank, we're collecting urine, plasma serum, CSF when applicable. So we're looking at some oxidative stress assays and tests to determine if there's any changes in the oxidative stress throughout the disease progression. So if there's any changes over time longitudinally. We're also seeing if there's any change in oxidative stress across those phenotypes that Dr. Patterson just touched on, if it was limb, if it's speech and swallowing onset, upper motor neuron, lower motor neuron, et cetera. And then also we're going to look at if there are any changes in oxidative stress between male and female as well. So they're on the up, and we're working real hard on those in the lab and staying busy through these times.
And also, the tissue repository contains DNA, and we share these specimens and, in fact, participate with the New York Genome Center to share our DNA because they're very busy sequencing the genomes of multiple subjects across the country. They have many thousands now of genomes from ALS subjects that they're looking at, and that's the way we can study the variability of ALS. In addition, we share our autopsy specimens with them as well. So we think that this is a huge resource to be shared with other investigators. And that really about wraps it up, but we wanted to let you know we're still busy. The lab is active. We feel very lucky and fortunate that we can continue our work, especially since the mice don't stop breeding and they need to be taken care of throughout all of this. So thank you very much for listening and joining. And if you have any questions, please let us know. Just send a quick email or a question to the foundation or directly to us at the lab. Take care, and thank you very much.
Thank you. So long.
Bye.
Thank you.